Okay, hello everyone. My name is Mengshi Zhou. I'm from Center for Artificial Intelligence in Drug Discovery at Case Western Reserve University. The title of my study is Combining Phenome-driven Drug Target Prediction with Patients' Electronic Health Records-based Clinical Corroboration towards Drug Discovery. So first, some backgrounds. The therapeutic dogma in traditional drug discovery is seek specific drugs to target individual genes for treating individual diseases. But complex diseases, such as neuropsychiatric disorders or cancers, they are associated with multiple genes and pathways. So the modulation of one single gene or one single pathway is often insufficient to accomplish a desired therapeutic effect. And growing evidence suggests that a drug exerts their effects by interacting with multiple targets in a complex system, like the right-hand figure shows. And the drug bank suggests each FDA-approved drug have 3 targets on average. Identifying of targets for FDA-approved drugs can open the door to new drug indications and accelerate multiple targeted drug development. But until now, the identified drug target interactions remains sparse because the experiments are often costly and time-consuming. So computational models have been developed to systematically predict drug target interactions. Computational models often predict drug target interactions by assuming similar drugs interact with the same targets or vice versa. This is just like how Facebook helps us find friends. So the more friends two people share, the more likely Facebook will recommend them to connect with each other. And similar situations for drugs. The similarity scores between drugs can be measured using different data. The most common used is chemical structures of drugs. For example, a previous study in bioinformatics applied a bipartite local model to predict drug target interactions based on drug chemical structure similarities and protein sequence similarity. And another example is using Gaussian interaction profile to model the drug chemical structure and genetic and genomic similarities for drug target prediction. The similarities can also come from drug genetic profiles. For example, some studies use gene expression profile correlations from CMAP to create a drug similarity network. And they assume drugs with a similar gene expression signature have the same targets. Recently, phenome data like drug clinical side effects or drug therapeutic information become more popular. Those data were observed from humans, so phenome-driven approaches are more likely to lead drug target discovery that can be translated into clinical application in the human bodies. And the most popular example for phenome-driven drug target prediction is a paper published in Science. So the authors calculated the similarity scores based on the number of the shared side effects, and they demonstrated that the identified drug target interactions can be validated in virtual binding assays. In our recent study, we developed a novel drug side effect context-sensitive network model to improve side effect-based drug target prediction. And compared to the traditional similarity-based model, our model can preserve more accurate information by connecting drugs directly through their shared side effect nodes instead of similarity scores. So for example, in this figure, we can see even for drug pairs that with similar number of side effects, shared side effects, our model shows that the side effects type they share are actually different. So we demonstrated the drug side effect context-sensitive network model had a significantly higher performance in drug target prediction as compared to the similarity score-based model. But our previous model only incorporated drug side effect information and genetic data. Other information like disease phenotype data may provide compensate drug discovery signals. So in this study, we first proposed an improved drug target prediction model, which we call it target predict, by incorporating additional human phenotype data that extracted from biomedical texts such as published literatures and clinical trials. We compared our target predict model with our drug context sensitive network model, and we demonstrated its advantage. We next combined the drug target interaction predictions from target predict with existing disease genetics to identify treatments for Alzheimer's disease, which is a neurodegenerative disease affecting nearly 6 million people in the United States. And we evaluated our identified drug candidates using patient electronic health records. So one of the clinical phenotype data we used is the disease comorbidity that we recently mined from 6 million patient health reports from FDA Adverse Event Reporting System. So basically, we extracted the patient reports and we used a frequent pattern growth algorithm to mine the disease co-occurrence patterns. So we built a disease comorbidity network, which includes 1,059 disease nodes and over 12,000 comorbidity edges. In our previous study, we demonstrated the disease comorbidity have correlation with identified or unidentified disease genetics. And another critical component of target predict is TreatKB, which is a comprehensive drug disease treatment relationship knowledge base that constructed from multiple free text documents such as FDA labels and published literatures. And in total, this knowledge base contains over 200,000 drug disease treatment pairs for over 2,000 drugs and 24,000 diseases. In our previous study, we demonstrated that the extracted drug disease pairs are strongly correlated with drug target interactions and disease genetics. So as shown in this figure, our target predict model incorporates over 850,000 phenotypic and genetic relationships among 24,000 biomedical nodes. So we have 3 layers on our model. The first layer is our drug side effect context-sensitive network model, context-sensitive network, which was built using drug side effect information from a database called side effect resources. And the second layer is our disease comorbidity network. The drug network and the disease network were connected using treatment pairs from the TreatKB knowledge base. And the third layer is a protein-protein interaction network, which include over 17,000 genes and their interactions downloaded from the string database. So we connected the disease network to gene network using disease associations from the OMIME database. And we connected the drug network with the genetic network using drug target interactions from drug bank. So the input of target predict model is a drug, and the output is a ranked list of each genes. To predict drug-target interactions, we extended a random walk algorithm to target predict. So given the input drug as the seed, the algorithm simulates a random walker who starts from the seed and estimates the probability of each gene being reached by the worker from the seed drug. So the update strategy of the probability vector is a Markov chain model. And the model updates the probability vector using the transition matrix, which represents the connectivity of the target predict model. So here, G represents the genetic network, D represents the drug network, and P represents the disease network. So the transition matrix include 3 inter-transition matrix of each network on the diagonal, and 6 inter-network transition matrix on the off-diagonal. For example, MDP represents the transition weight between drug network to disease network based on trait KB. We use the jumping probabilities to regulate the movements of the random walker, and we calculate our transition matrix. We use the de novo cross-validation to evaluate if adding those more information can help us have a higher performance. And we use the pre-session recall curves to show the overall performance. So on the left-hand figure is the comparison of target predict with our drug side effect context-sensitive network. So for target predict, we first connect the drug network and the disease network using the FDA-approved drug disease pairs from trait KB. So the green line here shows the performance of target predict, and the red line shows the performance of our previous model. So we can see target predict have a higher mean average precision and a higher precision at each recall level. And the pink line shows if we randomly connect our drug network and the disease network, our mean average precision will decrease significantly. So we also try to build a target predict by connecting drugs with diseases using the treatment pairs mined from post-marketing surveillance, which is more comprehensive but noisier than those extracted from FDA labels. So the right-hand figure shows, in this way, target predict, represented by the blue line, still have advantage compared to the drug context sensitive network model, but the improvement degree is lower. And we also demonstrate target predict have advantage for different parameter selection combinations. And those results together shows integrated disease information can significantly improve the predicting power of our model, but connecting the drug network with disease network use highly accurate treatment relationships is more important than using a comprehensive one. So after we demonstrated the advantage of the model, we further ask if the drug target prediction can be translated into clinical application. So we used our model to find new treatments for Alzheimer's disease. We downloaded over 600 Alzheimer's associated genes from AlzGene database. And for each drug, we predicted the probability of its targeted Alzheimer's associated genes. And we rank all the drugs based on the sum probability scores. So here we can see our top-ranked drugs include existing Alzheimer's disease treatments, and some of the drugs treat Alzheimer's disease risk factors like obesity. We performed a retrospective case control study to corroborate our Alzheimer's disease treatments. The electronic health records from Explorer's discovery platform was the data source in this part. And this database includes de-identified and population-level electronic health records of over 72 million unique patients from over 300,000 health institutes in the United States. And this database have heterogeneous patient information like their demographic factors, their disease diagnosis history, their medication prescription, and so on. And all the data were normalized using clinical ontologies from the United Medical Language System. So patients with a specific disease or use specific drug can be quickly extracted using their informatics tools. Since Alzheimer's disease is prevalent in elderly population with age over 65 years, so we use both Alzheimer's disease. So we use the patient with over 65 years old as the study cohort. And we use both Alzheimer's disease and dementia as the outcome, and the exposures are our top-ranked drugs. So for a candidate drug, we studied its association between Alzheimer's risk in patients with its original treatments. For example, if a drug treats type 2 diabetes, we evaluated in type 2 diabetes patients and adjusting for age, race, and gender. So the large electronic health records provide us enough sample size to do the adjustments. And we studied the top 30 drugs with enough sample size. And this figure shows among the top ranked drugs we studied, 6 of them showed potential clinical efficiency for preventing or treating Alzheimer's disease. For example, liraglutide is a type 2 diabetes drug, and it was associated with 24% lower Alzheimer's odds among the type 2 diabetes patients. And some animal model study can explain this observation because liraglutide can prevent the loss of brain insulin receptors and reverse the memory impairment in both mice and non-human primates. And all of our signals can be replicated if we using dementia as an outcome. So that's all the results I want to show you, but our study have some future directions. So first, we want to evaluate how our model performs in identify truly drug-off targets using experimental testing like animal models. And second, we want to make our model more comprehensive by adding different levels of data. For example, disease comorbidity data from a Medicare database have been built, which may compensate our disease network. And the other levels of data, such as gene expression and drug combination, can also help. Another future direction is our electronic health records are de-identified, so we cannot stratify time windows between the drug prescription and the disease outcome. So in the future, we want to evaluate our drug candidates using patient-level electronic health records. And our database, our network model, and the electronic health records study are general and flexible. So in the future, we want to apply this target predict model to identify treatments for other diseases like depression. So my research was funded by multiple institutions, and I thank you for all your attention, and I'm happy to answer any questions.